Torres-Dowdall. I'm a postdoc at University of Constance in Germany. And today I want to tell you a little bit about the work we're doing in the molecular evolution of con-opsin genes in cichlids. As a quick reminder, opsin genes code for the protein component of photosensitive pigments in the retina of animals. The other component is photosensitive chromophore, and the interaction between these two is what determines the sensitivity of the photosensitive pigment at different parts of the light spectrum, which is usually characterized by the peak of maximum absorption. Here it's 534 nanometers, which characterize the green photosensitive pigments in humans. What is great about opsins is that we have a very good understanding of the phenotypic consequences of molecular changes. For example, we know that the underlying molecular mechanisms that result in the differentiation between the red and the green photosensitive pigments in humans is due to 3 amino acid substitution in the binding pocket of the chromophore protein. But we also know that differential expression can affect the phenotype, and this example to use is color blindness in humans, because we understand it quite well. So failure to express either of these two con-opsins will result in a problem to differentiate between greens and reds. And of course, in humans, this is not necessarily adapted, but it's an important mechanism in organisms that have multiple genes, con-opsin genes. And fishes are one of these cases. In particular, cichlid fishes. Cichlids are really interesting group to study because they are one of the largest families of vertebrates with more than 2,000 species, most of which live in Great Lakes in Africa, and where they have undergone these splendid radiations where they diversify around many phenotypic aspects. One of those you can see here, it's coloration. And this has been interpreted as selection in intraspecific communication, so male-male competition or mate choice. And of course, it assumes that they have a receptor that can sense this difference in coloration. And actually they do, they have a splendid visual system. So compared to humans that only have 4 opsins, cichlids have 8 opsins. One is rhodopsin, which is sensitive to, it's responsible for vision under dim light conditions, and then 7 con-opsins that are responsible for color vision, and their peaks of maximum sensitivity span much of the visual light spectrum. So today I will focus on con-opsins. So the way they take advantage of this diversity in genes is not by expressing all of them at the same time, but rather expressing different subsets in different environments. For example, in clear water, cichlids express a short subset that is composed for those opsins that are sensitive to more short wavelength, while in more murky waters, they express a longer subset, which is composed by opsins that are sensitive to longer wavelength. Another way they can adapt to the environment is by structural changes in protein itself. And the Pulmonamyla from Victoria are a good example. So this is the light spectrum in Lake Victoria, and you can see it change very rapidly as you go deeper and deeper. These are different depths, and Pulmonamyla fish that live in these different depths differ in the frequency of 2 alleles of the long wave sensitive pigment, opsin pulmonamine, that have different peak of sensitivity, and they differ in a way that is adaptive. And both of these mechanisms have been linked to the diversification rate in cichlids. And especially this one has been mechanistically linked through sensory-derived speciation. So our current understanding is that the visual system of cichlids play an important role in their adaptive radiations, and that can explain some of the amazing diversity we see in African cichlids. Of course, there are other lineages in cichlids that didn't diversify that much, so we can use it to compare it. And there are 2 main lineages here. The more basal one is composed by cichlids from excuse me, Madagascar, Sri Lanka, and India. And the sister lineages to African cichlids are the Neotropical cichlids that underwent some radiation, but not as splendid as those seen in African cichlids. So for the rest of the talk, I will focus on the comparison between African and Neotropical cichlids. And unfortunately, we don't know much about the visual system of Neotropical cichlids. So as a reminder, these were the 7 conopsins found in African cichlids, and a pioneer work done in Belinda's Young lab identified only 4 of those genes in Neotropical cichlids, finding that the other ones were either not present or became pseudogenes. So this has important consequences because it reduced the ability to express different genes in different environments and probably the ability to rapidly colonize different life conditions. Another difference between African and Neotropical cichlids is in the rate of molecular evolution of some of their genes. And for example, we found this different in rhodopsin, the one that was responsible for vision under dim light conditions. And we found much faster rates of evolution in African cichlids compared with Neotropical ones. And we thought that these were due to several factors, including ecology, demography, and the evolutionary history of the protein. But what we don't know if this is characteristic of all opsins or just rhodopsins, so I wanted to explore if this is also common in conopsin genes. So my general question is if this difference between African and Neotropical cichlids are real, and if those can explain difference in the rate of molecular evolution. And for that, I have 2 specific questions. One is if the lower diversity seen in Cranecycla frenata is characteristic of all cichlids, Neotropical cichlids, or just that lineage, and also if there are differences in the rate of molecular evolution in conopsins, as we saw in rhodopsin. So the first question was, is this low genetic diversity seen in Cranecycla characteristic of all Neotropical cichlids when you compare it to the African cichlid? And I was lucky that we have the draft genome of Aphylophis cichlids in Mayo Lab, so I was able to find the seven conopsins in this genome, and these are autologous to the genes found in African cichlids. And then I actually use this as template to design primers to amplify the different members of the Neotropical cichlids that are representative of the 3 major subfamilies within Neotropical cichlid. And what you can see is that the 7 conopsins are present in most of the families. And yes, sure, it became pseudogenized in several lineages, but overall you can see that the 7 conopsins are present across the phylogeny. What is very interesting actually is this, the 2 parallels of RH2A, alpha and beta. This is a duplication that was thought to be specific to African cichlids, but that is in disagreement with the fact that we find these 2 parallels in the 3 lineages. Here it's Africans, here it's Neotropical, and this is Paratilapia from Madagascar. So we find that the 2 parallels in the 3 lineages, but when we make a gene tree, what we find is a topology that suggests that these underwent duplication event 3 times as the 2 parallels clustered together within each of the lineages and not across the lineages. But a different explanation for this is that it could be gene conversion. And actually I tested for that with several different tests, but just to give you an example, this is Garin Hi-Fi, and what you find is that significant evidence of recombination or gene conversion, and there is a breakpoint that separates 2 segments that have completely different topologies. So if we make trees with these 2, what you find is that in the first segment, which corresponds pretty well with the first exon, you recover the topology where it suggests only 1 duplication event occurred between these 2 paralogs, and you can see that they are clustering together, the 2 lineages clustered together within each paralog. If you use the second fragment, what you recover is a very complicated tree that suggests multiple duplications or actually a strong signature of gene conversion within each major lineage of Neotropical cichlids and even some lineages of African cichlids. And an interesting case of this gene conversion is our Cranecycla, which are these guys here, where both parallels cluster together. And actually they are 99% similar, which suggests that gene conversion might have played a higher role in this particular lineage, resulting in the loss of functional diversity in this particular gene. My second question was if there were differences in the molecular rate of evolution between these 2, between the African and Neotropical lineages. And the first thing I use is compare the test for positive selection within each of the lineages, African and Neotropical, for all the different conopsins. And what I found is that some opsins are under selection, other ones are not. But overall, what you don't see is that there are no really big differences between the two lineages. So some genes are under positive selection in both lineages, and other ones are not under positive selection in both lineages. But this does not compare the rate between lineages, so I used clade models where I tested the difference on the fit of the model that considers just all cichlids together versus another model that allows for variation in the rate of molecular evolution between the two lineages. And what I found is that not much difference there either, except in the long wave sensitive pigment and the short wave sensitive pigment. And this is actually really interesting because these are the two genes that are at the extremes of the light spectrum. And those two extremes, if you have to adapt to light conditions there, you cannot do it by expressing different opsins because there is nothing else outside, so you have to adapt by structural changes. So the fact that we find different rates between the lineages and the two extremes is quite interesting. So to sum up this, what I found is that the low gene diversity that we saw in green cichlids is not characteristic of all neotropical cichlids, but gene conversion or pseudogenetization might have affected functional diversity in different lineages of neotropical cichlids. Also, what I found is that there is no much evidence for heterogeneity in the rates of molecular evolution between African and Neotropical cichlids, except for some genes that might be really interesting and worth exploring a little more. So, to answer my main question is that I don't think there are differences in the visual system between Neotropicals and African cichlids that can explain the difference in diversification rate between the both lineages. I'm not saying that the visual system does not play an important role. What I'm saying is that you cannot explain the difference between the two lineages. But probably it did play an important role, and in neotropical cichlids there are certain lineages that diversify along coloration axes as well. And with that I finish my talk, and I thank you for listening and my funded sources. And also if you are interested in plasticity, cichlids or opsins, or the interaction with them, you should check the Torres-Dowdall this afternoon.